Thank you. Thank you. Good morning, ladies and gentlemen, and welcome to my lecture, which is entitled as Recent Developments in Molecular Imaging. Okay, my voice is clear. We will start here for recent developments in molecular imaging. First of all, let us define what is molecular imaging. Molecular imaging, it's the in vivo visualization, characterization, inspecting, and measurement of biological processes, but at the cellular and molecular level. Let us say what do we mean by measurement? Measurement. Molecular imaging can give us qualitative and quantitative measurement, as well as it will image for us step by step the basis of a disease rather than the end stage of molecular changes of a disease. Let us say we will take an example of this disease. We can say cancer. Molecular imaging will allow us as clinicians or technologists or researchers to not only see where is the location of this tumor if the patient has a cancer, but we can visualize it through the molecular imaging. Molecular imaging can give us the way to see the expression, the activity of specific molecules such as proteases, protein kinases, step by step. It will tell us how the disease was developed. And you can see here, step by step, you can see the activity or the expression of any receptor or any protease or protein kinases in the process of developing a disease. It will show us as a molecular imaging. Also, molecular imaging can visualize for us the biological process. Okay, let us take an example, which is apoptosis. Apoptosis, we say apoptosis, like the definition of apoptosis, it's a programmed cell death. So if we have a healthy cell, it will be developed after some time to apoptotic cell. The molecular imaging can give us an idea about step by step the development of this disease. So you can see here how the development of cancer, for example, the DNA damage, and so on, receptors or messengers or kinases, or the function itself of apoptosis or biological process itself, how we develop this cancer. So just in brief, this one. Then we can get from all of this information a very good message of molecular imaging, which is saying for us that we can early detect cancer, we can individualize or personalize the treatment of cancer, and we can develop a drug as well as understand how this process of cancer arises. There are many diseases emerge us, let us say that some diseases give us the idea to emerge the molecular imaging. The diseases like cancer, heart diseases, and brain disorders. Those diseases emerge the molecular imaging and necessitates to develop more molecular imaging. So molecular imaging, again, as a definition and its advantage, it will give us the benefit to early detect, understand, and manage and see the effect of therapy of a disease. Now, the molecular imaging has many advantages over the traditional in vitro imaging. We can say that molecular imaging is non-invasive, while the traditional in vitro, it's an invasive. We can take cells during the process of imaging or during the process of in vitro study. So molecular imaging, it can give us also dynamic process. It will trace movement of cells, and also traditional in vitro in the opposite, it will give us static image, and we cannot interrogate any process or the signaling process. It cannot be interrogated. In molecular imaging also, it can give us rapid pharmacokinetics and rapid pharmacodynamics, means that the molecular imaging, it will save time. What is the definition of pharmacokinetics? And what is the definition of pharmacodynamics? If we want to define them, pharmacology or the drug itself during molecular imaging, we can detect its effect with time. So we can say pharmacokinetics. While if we want to detect the effect of pharmacology or a drug with a dose, we can say pharmacodynamics. So molecular imaging will give us an idea. Better than the traditional in vitro. In vitro, we cannot do anything about this. Also, molecular imaging, we can do a repetition of animal studies in the same animal itself. So we can save time, save animals, and so on. While the traditional in vitro, we cannot do this. Also, molecular imaging, it can provide us with temporal and spatial resolution. With the new development of modalities, we can get more adequate temporal and spatial resolution. And this is not available in the traditional imaging and the traditional in vitro imaging. So, again, this slide will summarize for you what is about the recent developments of molecular imaging. So we can say that non-invasive molecular imaging technologies will give researchers and anyone to study small animal models for human diseases, and we can, along with the improvement of instrumentation and along with the better identification of imaging targets or primers or studies, we can say that along with the continued improvement in the instrumentation and the better imaging targets and specification of targets, along with the genome-based approaches and the better innovative chemistry, we can get the best idea about molecular imaging, and we can disease diagnosis and therapy, we can get it. Okay. Recent developments in molecular imaging, I can say we can conclude this in three things. Introducing novel agents, second, signal amplification methods, and developing new imaging technologies. Once we get such things, we can get recent developments in molecular imaging. Let us talk about introducing novel agents. Okay, molecular imaging agents, we can say that we have many molecular imaging agents, but we can take some examples. I will not take all of these examples, but we can talk about some examples. Small molecules, we have peptides, we have antibody, we have aptamer, and antibody and nanoparticle. Small molecule, one example is fluorine 18 radiolabeled with fluorodeoxyglucose, F18FDG. It's a small molecule, and all of you know the benefit of F18FDG. Okay, so it will image some small molecule, it will image some parts or processes in the body. Let us talk about the peptide. We have an example which is RGD peptide. It can be labeled with a fluorescent peptide. It's an imaging agent, and we can get this imaging agent. See, this is a peptide. So we can get it through the process of cancer or through the process of myocardial infarction, and we can label the RGD peptide. Okay. Sorry. This we can label the peptide itself with an imaging agent. We can get the process of molecular imaging, and we can tell what is happening in this disease. For example, the integrin, which is alpha V beta 3, it's an imaging agent. We can tag it with F18FDG, and we can image a process of myocardial infarction. So we can see the expression of this myocardial infarction in molecular basis. Also, we have a primer or a target for molecular imaging. It's called antibody. Antibody, it's an engineered protein, small size amino acids. We can tag it with beta agent, gallium-68, and we can get it through a process with tagging with antibody, fluorescent antibody, and it will image some receptors in the cancer. We can say human epidermal growth factor receptor type 2, and we can image it. These are small examples of molecular imaging agents. all of these, it will conclude for us the development of new or novel imaging agents in the molecular imaging. So here we can say that antibody, for example, we have a study in nude mice. We xenografted some tumors, okay, and we can get the molecular imaging through using the antibody. And you know that in the antibody, what was the definition of antibody? Again, I said antibody is an engineered small protein, small size amino acid. So this is a benefit of molecular imaging. You can see this small engineered protein give us an idea about the tumor process molecular imaging. But after, and you can see here the panel of A, it is the tumor xenografted here in the nude mice. But after treatment with certain drug, we can see the effect of this process of treatment using this antibody. It's the speaking that it is a molecular imaging technique. So we can say here panel A is the imaging of a tumor. Okay, after treatment you cannot see the tumor. Okay, but the process is molecular imaging using antibody, which is a novel agent in molecular imaging. Also, we have another agent for molecular imaging. We can say its name aptamer. It's defined as single-stranded DNA or RNA oligonucleotide. We can say it is similar to an antibody, replacing the antibody, but it's fast and good. It has a good specificity and selectivity. So we can image all of the processes of molecular imaging through using aptamer, which is a single-stranded DNA or RNA. So for example, in therapy, they have used the first aptamer for the treatment of macular degeneration, and it is used in treatment and in investigating certain diseases following the steps of molecular diseases. Okay. RNA aptamer, it's an antibody-like properties, as I said, and we can get benefit from this novel agent for molecular imaging. See here in this nude mice, we have a nude mice. It has xenografted some breast cancer, and you can see here if the aptamer is binding, you can see it is shown. But if the aptamer is not binding, you can see it is not shown here in this example. So the benefit of introducing novel agents will give us more ideas about what is happening as a basis of a disease for molecular imaging. Okay. Now we will talk about second one, which is the signal amplification methods. We need to amplify our signals during imaging. This is the second criteria or second need for recent developments in molecular imaging. So let us say that we have PET imaging, we have SPECT, CT, and MRI, ultrasound. All of that is known since the 80s. Okay, we introduce more facilities or more technologies, optical imaging. Okay, but we need to amplify our signals and increase it. For example, we have positron emission tomography, PET. We can introduce iterative image reconstruction and correction methods. Okay. Okay, this will play around the amplification of signal. So iterative image reconstruction and correction methods. This was introduced as a new amplification signal. And also new scintillation materials were introduced recently, so it will increase the signal in the PET imaging. And it will compensate for depth interaction effects, and we can see more structures in a physiology or in a functional way, but better than before. And it will help us as new developments for molecular imaging. So Also, as an example, the magnetic resonance imaging, MRI systems, we have to improve radio frequency call technology, and we have to get higher field strength. And this is going on, and it is one of the current or new signal amplification in molecular imaging. Actually, molecular imaging is the same as nuclear medicine techniques, but with deeper and with better advantages and better effect. So now, after recent developments in molecular imaging and introducing novel agents and signal amplification methods, we will talk about new or developing new imaging technologies. يعني plus the presence of X-ray and CT, MRI, ultrasound, or PET CT, fused PET MRI, all of these systems were present, but with the new developments in signal amplification and so on, we will get next generation of systems. It will deal with molecular imaging, therapeutics, and diagnostics. We can say it is emerging image-guided gene therapy. As I said, the technology will depend on genome-based approaches and also on the innovative or new chemistry. So we should take care about all of these aspects, and we collaborate all of these techniques, and we put it in a new imager. This table is showing some advantages or some parameters for each technique, like CT, MRI, PET, and SPECT, ultrasound, plus that optical fluorescence imaging, optical bioluminescence imaging, and surface enhanced Raman scattering, and intravital microscope. All of these has their own benefits. The most important is to get more spatial resolution, more sensitivity from these machines or from these techniques, and also to combine the best one to get the image in a clearer way. So it will provide us for best signal amplification and best quality of answering the question of a disease. Let us say for PET and SPECT, and if you combine it with, for example, the smart MRI, you can get the benefit of each one with its advantages. Like sensitivity, we can get PET in this range, okay, along with the MRS, which is the highest sensitivity. And also spatial scale or spatial resolution, we can image certain small animals till the human, but we can get limitation of spatial resolution. So you will take care about the advantage of each one, and you combine and infuse each technique in order to get your image. So this is the play around of molecular imaging. We have new also techniques. We can collaborate with other sciences like physics, like biology, like chemistry, and so on, in order to get our imaging of molecular imaging. So the PCR polymerase chain reaction, I do it in my lab also in this time. So we can do a PCR polymerase chain reaction in order to get novel answers to certain questions of a disease. So we have to amplify some single DNA, a piece of DNA, you know the DNA, okay? And we can get some certain particular DNA sequence and play around in the genes and so on, and we can get an answer for the diagnosis of a disease, for example, okay? This is this machine here. I have it in my lab. It is the real-time PCR machine, and it has a software, and I can get relative quantitation of gene expression analysis, and it will give me some idea about the expression of certain diseases, okay, in a certain group. So, for example, the recent development of methods for gene to visualize the gene expression we can hybridize it with arrays of DNA, and we get some certain information. And we can compare the patients whether they have this disease in a tumor or certain, certain gene is overexpressed in a tumor or not. So we can get an idea about this. This is also the basis of molecular imaging. So if we have gene assay, okay, we can predict the study or predict the certain parameter in a disease, and we can detect it, and we go from molecular diagnosis to molecular imaging. We can image it through our agents that I said, like antibody, small molecules, aptamer, and so on. So we can image it, we can get molecular imaging, and then we can prevent it through using the idea about this disease. So molecular therapeutics, all of this will combine together and will give us in the future the individualized medicine. It will tailor the medicine for a certain patient only, and it will give us an idea about this, and it will prevent the disease also. So in the methodology, we can use preclinical studies using small animals models, okay, mimicking the disease of a human. And for new technology for imaging, as I said, we have CT, but with micro, also microbit, microspect, and micro MRI, and we can combine all of these techniques with microspect optical reflectant or reflectant fluorescence, using some fluorescence or luminescence targets, and with optical bioluminescence we can get some idea about the disease. Also, we can translate all of these preclinical studies to human beings, and we can also ask about some new agents or new specification of disease or new specification for BET studies. Let us say for BET we should specify the targets or specify the radiopharmaceutical. Let us give the BET radiopharmaceutical more specification. For example, F18 FDG is not specific, but we want to develop some targets of BET radiopharmaceuticals to be more specific. So we will do some tumor metabolism with another agents which should be specific to detect that metabolism. Proliferation, apoptosis, hypoxia, and angiogenesis. All of these will be leading toward specifications of these agents. So if we combine it with the new technologies, we will get better idea or picture about the disease. Again, we can combine all of the diagnostic imaging, as I said, with the diagnostic pharmacology and with the genetic based approaches, and we can get also molecular imaging. It's all together the same thing, but collaborating with each science. Genomic development also, with the disease biomarker questioning and finding some biomarkers. If we combine them with targeted chemistry and with imaging technology, not only we can get imaging of molecular, also we can get preventive medicine, and we can personalize this medicine to certain patient. Okay, now the future of this molecular imaging has many challenges. Okay, the same thing of the development of new imaging technologies and the new novel agents in molecular imaging, we have some challenges. We should find out more imaging agents, biomarkers, and we should create more chemistry, as the same as the need of nuclear medicine. We should develop instrumentation, and we should look at more multi-modality trained staff, and we should get more clinical translation of a disease, and we should get more multiplexing capabilities and sensitivity of all of these agents and all of these technologies. And also, we cannot forget the cost and availability of these facilities. This is what all about the molecular imaging, and I thank you for your attention. Thank you.